We were going all the way back to the very beginning, tracing the formation of the human nervous system from, a simple flat sheet of cells into the complex thinking machine that allows us to even have this conversation.
It's the blueprint of thought itself.
It is. This is the architectural blueprint.
And our mission for you, the listener, who wants this rapid but really thorough knowledge, is to synthesize a challenging stack of sources. We're focusing entirely on the embryogenesis and maturation of this system.
Yeah.
We're not just listing parts here. We're trying to understand the foundational why. Why is human neurological structure the way it is? What makes it function and what makes it so vulnerable?
And when we talk about where it all comes from, the origin, we're really talking about two primary sources.
This is the critical starting point.
You've got the central structure, the neural tube, which becomes the CNS, and then you have these scattered, specialized components of the ectoderm that build out everything else.
Everything else. The PNS, the sensory organs.
Yeah.
And that structural duality is where we have to begin. Our sources really underscore that this isn't just passive growth.
Yeah.
It's an intensely dynamic construction project. You have precise cell migration, complex signaling gradients, and interactions across multiple germ layers. The ectoderm provides the bulk, for sure, but the mesoderm dictates the fate of the spinal cord. Even, get this, the oral ectoderm plays a surprising role in building the brain.
Wow.
So we have to track this chronologically, piece by piece, from the simplest fold to the final myelinated structure of the newborn.
So let's begin where it all starts, with that initial folding process. It's called neurulation. And it sounds simple, like rolling up a carpet.
It does.
But the sources immediately make a distinction between two fundamentally different methods: primary and secondary neurulation.
Right. And primary neurulation is the one most people think of. It's the process that builds the main rostral body, so the head and the upper trunk.
And this happens early.
Very early and very fast. It kicks off around stage 9, which is about 18 post-fertilization days, and it's pretty much done by stage 12, so day 26. This process establishes the fundamental geometry of the entire central nervous system.
So walk us through the mechanics of that. You start with this flat neural plate. What happens?
Well, the neural plate, which is just this thickened area of the dorsal ectoderm, it begins to lengthen and broaden out dramatically.
Okay.
Then its lateral edges start to elevate. These become the neural folds, and they flank this central depression, the neural groove.
So you have these two ridges forming.
Exactly.
Yeah.
And these folds, they just keep elevating, they curve towards the middle, and eventually they meet and fuse right in the midline.
And when they fuse?
They've effectively pinched off a hollow tube, the neural tube, from the surface ectoderm that's left on top.
Now, you mentioned this fusion is a critical sort of bidimensional process. It's not happening all at once along the whole length. So where does this zipper actually start?
That initiation site is absolutely critical. It typically begins in what will be the posterior cervical region.
Okay, the neck.
Right, corresponding to the level of the 4th to 6th somites. And from that starting point, the fusion, the zippering, proceeds in two directions at the same time.
Rostrally and caudally.
Exactly. It zippers rostrally up toward the future brain and caudally extending down toward the tail.
And the importance of this is that for a short time, you have two openings, the neuropores.
Precisely. The whole goal of primary neurulation is to close both of them, the anterior or rostral and the posterior or caudal neuropores.
Right.
Their closure establishes the completely internalized fundamental CNS structure. The anterior one closes first, usually by day 25, then the posterior one a few days later, around day 28. If either of those steps is delayed or incomplete,
You have profound consequences.
absolutely.
So primary neurulation sets up the brain and most of the spinal cord. But for the very tail end, the process changes completely. That's secondary neurulation. Why the change in strategy?
Well, the caudal region, it just requires a totally different mechanism. Part of it is because of the structural constraints down there, and part of it is the cellular components that are present.
So it starts after the first process is done.
Right. It kicks in around stage 12, focusing specifically on the area just caudal to where that posterior neuropore closed.
So how is this different from the folding we just talked about? It's not a zipper.
Not at all. Instead of folding surface ectoderm, secondary neurulation involves the aggregation of this dense cell mass. It's called the caudal mass.
A solid block of cells.
A solid block of cells. It's made of caudal mesoderm and deep caudal neural crest cells. This aggregate then undergoes a process called cavitation.
Cavitation. So it hollows out from the inside.
That's it exactly. Vacuolas, little spaces, appear inside the mass. They merge, they coalesce, and they form a central lumen, a central canal.
And then it just connects.
Once that lumen is established, The whole structure just fuses with the caudal end of the primary neural tube that was already formed. So it's a method of condensation and hollowing, not folding and zipping.
Two completely different methods building one continuous structure.
Mm-hmm.
If we're looking at an adult, where's the dividing line? What should we remember?
The sources are very clear on this. The part of the spinal cord from primary neurulation goes down to about the level of 20 somites. That's roughly the upper lumbar region.
Okay.
Secondary neurulation forms everything below that. So generally that corresponds to the L4, L5 level and the very tapered end, the coronis medullaris.
And this dual origin probably explains why you see certain types of anomalies in certain regions.
It does. And because this whole process of folding and fusion is dictated by such precise timing and mechanics, any failure leads to developmental catastrophe. We call these failures dysraphism.
Right. Dysraphism is the technical term for when the tube just fails to close completely, leaving it open.
Yes, leaving it open to the dorsal midline. And these conditions are directly linked to those failure points we just discussed.
So let's start at the head end. What happens with a rostral failure?
A failure in the rostral midline, where the anterior neuropore should close, often results in anencephaly.
Where the brain vault just fails to develop.
The vault fails, the brain tissue is severely compromised. Or you can have craniocatechesis, which involves both the brain and the spine. In either case, the lack of proper covering and development is tragically incompatible with life.
And then there's the more common, though widely varying, failure at the caudal end.
And that's spina bifida, the failure of the neural tube to close in the caudal region. The severity here depends entirely on the level and the size of the defect.
It can be quite mild.
It can. Failure of just the bony arches to close, spina bifida occulta, is the mildest form. But if the neural tissue itself protrudes through that defect, which is a meningomyelocele, the neurological consequences are severe.
And this caudal failure is often complicated by a structural problem much higher up in the brain.
Yes, the vast majority of severe spina bifida cases are complicated by something called a Chiari-II malformation.
How does that work?
Because the spinal cord is anchored, it's tethered down low, it changes the pressure dynamics inside the skull. This causes the hindbrain, specifically the cerebellar tonsils and the medulla, to be pulled down through the foramen magnum into the spinal canal.
So it's literally pulling the brainstem down.
It is, and this can lead to hydrocephalus and extensive neurological dysfunction because the brainstem itself is being compressed. It just shows how a failure at one end of the tube can create massive structural problems hundreds of segments away.
Okay, so if the neural tube forms the CNS, we have to introduce the cells that build literally everything else, the entire peripheral nervous system and so much more.
The neurocrest cells.
Or NCCs. They've earned the nickname, the fourth germ layer.
They absolutely deserve that title. Their origin is unique. Their migratory ability is legendary. They originate from the neuroectoderm right at the lateral edges of the neural plate.
Right at the border.
The boundary between what will become the neural tube and what will remain as surface skin.
And to migrate so far, they have to undergo this radical transformation.
They do. It's called the epithelial to mesenchymal transition, or EMT. Now, epithelial cells are normally stuck together, tightly bound.
Right.
To migrate, the NCCs have to temporarily lose that adhesion. They do this by downregulating adhesion molecules, like E-cadherin. This lets them break free from the neural fold and become mesenchymal.
Meaning they're loose, mobile, migratory cells.
Exactly. And that EMT process is the key that unlocks their potential.
And once they break free, I mean, the list of where they go is just staggering. We know they form the PNS sensory and autonomic neurons.
Yeah.
But what about those non-neural contributions that make them so famous?
The list is immense. I mean, they populate mesenchymal tissue, so they contribute to bone, cartilage, and dermis, especially in the face, the jaws, the neck. They form all the melanocytes, the pigment cells in our skin. They give rise to many endocrine cells, and critically, they form the chromaffin cells of the adrenal medulla.
And chromaffin cells are essentially modified post-ganglionic sympathetic neurons, aren't they? They pump adrenaline straight into the blood.
That's exactly right. And that lineage link ties the acute stress response, the fight or flight response, directly back to these migratory NCCs.
So my facial skeleton, my skin color, and my stress response all share this common, highly mobile embryonic origin.
It's incredible architecture.
That is fascinating. Now, given that they spread through the entire body, their pathfinding has to be highly controlled. How do they know where to go? And maybe more importantly, where not to go?
Their migration is strictly segmental. It's tied directly to the somites, those blocks of mesoderm that define the body's segmentation, and the sources detail a very precise control system that defines their path.
Okay, let's look at the main path for forming the ganglia, the ventral pathway.
Most of the NCCs that are destined to form the sensory ganglia, so the dorsal root ganglia and the autonomic ganglia, follow this ventral route.
Yeah.
And crucially, they move ventrally, but they only migrate through the anterior half of each somite.
Why do they strictly avoid the posterior half? What's going on there?
It's a fantastic mechanism of repulsive signaling. There are factors, like ephrins, that are expressed in the posterior half of the sclerotome, the part of the somite that will become the vertebrae.
A keep-out sign.
A molecular keep-out sign. The NCCs carry the corresponding F receptors. When they bump into the posterior half, the repulsive signal literally forces them into the anterior half.
And that ensures perfect segmentation.
It ensures that the spinal nerves and the sympathetic chain ganglia are perfectly segmented and aligned correctly with the developing vertebrae. Without that repulsion, the system would just be disorganized chaos.
And the melanocytes, the pigment cells, they take a completely different route.
Yes, they follow the dorsolateral pathway. So instead of diving down ventrally through the somites, these NCCs migrate between the somite and the surface ectoderm.
So their destiny is determined by the path they take.
The path they take and the local environmental cues they receive when they get there. That's what decides if they become a sensory neuron or a pigment cell.
And there's a powerful clinical correlation here related to this whole signaling and pathfinding system.
Oh, the integrity of this NCC migration is absolutely vital. Disruption of these pathways is implicated in numerous conditions we call neurocrystopathies.
Right.
And the source specifically cites Waardenburg syndrome, which often involves abnormalities in skin and hair pigmentation, that's a melanocyte issue, and sometimes congenital hearing loss, which is linked to inner ear structures that are partly derived from NCCs.
And that's tied to a specific gene.
It's tied to issues with the PAX3 gene, which is essential for NCC survival, for their fate specification, and for their movement. If the machinery that controls the migration fails, the consequences are body-wide.
So we have the CNS from the tube, the widespread PNS from the neural crest. But the sources point to a third, highly specialized contributor, especially for our senses.
The ectodermal placodes.
What exactly is a placode?
Placodes are these localized thickenings of the surface ectoderm that appear up near the front, near the closing neural tube. They're highly specialized little patches of tissue dedicated to sensory input.
So which specialized sensory structures rely entirely on these placodes?
We can break them down by region. So you have the nasal or olfactory placodes. They invaginate to form the specialized epithelium that gives rise to the olfactory nerve, cranial nerve the first.
Okay.
Then you have the lens placodes for the visual system. They're induced by the optic vesicles, and they invaginate to form the lens vesicle, which becomes the lens of the eye.
An ear.
And finally, the otic placodes. They invaginate very deeply to form the otic vesicle, which is the precursor to all the intricate structures of the inner ear for hearing and balance. Cranial nerve 8.
So those are the discrete organs. But where it gets really complicated is that the placodes also cooperate with the neural crest to build the sensory ganglia for the major cranial nerves.
That's where the concept of neurogenic placodes comes in. Specific placodes, like the trigeminal and the various epibranchial placodes, they contribute neurons directly to the cranial sensory ganglia, specifically V7, NEET, and X.
This is our classic aha moment.
Yep.
Many cranial sensory ganglia are not single source structures from the neural crest. They are composite.
They are absolutely composite. The sensory ganglia of cranial nerves V7, NEET, and X are dual origin structures.
So what does each part contribute?
The neural crest generally contributes the general somatic afferent, GSA, neurons, so general touch, pain, temperature sensation. But the ectodermal placodes, they contribute the special visceral afferent, SVA, neurons for things like taste, and sometimes the general visceral afferent, GVA, neurons for organ sensation.
So this dual input lets those ganglia process a much wider range of information.
A much wider spectrum reflecting the really distinct functional needs of the head compared to the trunk.
So in the trigeminal ganglion, CNM, the neurons that feel a scratch on your face come from the neural crest.
Right.
But the specialized neurons that are responsible for taste, which are mediated by 7-IXNX, those come from different ectodermal plicodes.
That's it exactly. It's this integrated system where the NCCs provide the structural framework and the general sensory function, and the plicodes come in and provide the specialized sensory acuity that really characterizes the head region. This integration is why cranial nerve development is so much more intricate than a simple spinal nerve.
Okay, now let's move rostrally and shift from the simple tube to the development of the brain itself. The rostral neural tube swells and segments very, very rapidly, establishing the core regions of the brain.
By stage 11, which is around day 24, we see the foundation of the brain laid out in what's called the three-brain vesicle stage. These three primary swellings establish the three major functional divisions.
And those are?
First, you have the prosencephalon, which is the forebrain. It's responsible for higher cognitive, emotional, and regulatory functions. Second, the mesencephalon, the midbrain, which serves as a crucial relay and reflex center. And third, the rhombencephalon, or hindbrain, which is responsible for vital survival functions and coordination.
But the head is small, and this tube is expanding so fast. So even at this early stage, it can't be straight. It has to start folding on itself. These are the flexures.
The flexures are an architectural necessity. The first one is the cephalic flexure. It forms in the midbrain region, and it bends the forebrain and midbrain ventrally, sort of forward toward the chest.
Right.
This aligns the brain axis a bit orthogonally to the spinal cord. The second is the cervical flexure, which forms at the junction of the hindbrain and the spinal cord. These initial folds begin the process of compacting the whole developing brain structure.
That three-vesicle structure only lasts for a few days before it rapidly refines itself. So by stage 13, day 28, we hit the five-brain vesicle stage.
This is the critical subdivision stage. The prosencephalon divides into the telencephalon, those are the two lateral cerebral hemispheres, and the diencephalon, which is the central thalamus and hypothalamus. The mesencephalon remains a single structure, and the rhombencephalon divides into the metencephalon, which becomes the pons and cerebellum, and the myelencephalon, the medulla oblongata.
And this subdivision introduces a third and structurally very disruptive flexure, the pontine flexure.
The pontine flexure is functionally critical. It causes this localized dorsal bend at the junction of the metencephalon and myelencephalon.
So it bends the other way.
It bends the other way. And unlike the first two, this dorsal bend acts like a hinge, forcing the walls of the neural tube in the brainstem region to open up and spread laterally.
Like opening a book and laying it flat.
Exactly like that. And the spreading is the reason why the brainstem nuclei are organized in columns along the floor of the fourth ventricle, instead of in those compact horns like we see in the spinal cord. It fundamentally changes the architectural layout.
Let's focus in on that rhombencephalon, the hindbrain. This area is responsible for organizing all the cranial nerves and our vital reflexes. And it segments into these 8 transient repeating units called rhombomeres.
The appearance of rhombomeres R1 through R8 is a visible manifestation of the molecular segmentation that's happening underneath. They're clear by stage 12, and they persist through stage 18. And their significance is profound. They are the foundational organizational centers for the cranial nerve nuclei.
And the migration of their associated neural crest cells.
Exact.
So they're not just anatomical bumps, they are destiny maps.
Yeah.
How is their identity determined?
This is governed by one of the most remarkable conserved mechanisms in all of biology, the segmented expression patterns of Hox genes.
Ah, the master control genes.
And master control genes. The rhombomeres represent a form of serial homology. They're repeating, segmented units, just like vertebrae or somites. And the specific combination of Hox genes expressed in each rhombomere acts like a genetic address defining its fate.
Can you give us an example of how that Hox code works?
Certainly. The sources show that specific combinations of Hox genes are expressed in alternating rhombomeres. So, for instance, HOXA2 and HOXB2 are typically expressed in rhombomeres R3 and R5. While HOXA3 and HOXB3 are expressed in R4 and R6, these combinations dictate which cranial nerves will form their motor nuclei there, and critically, which neural crest populations will migrate out to form the associated ganglia.
So if you were to experimentally mess with the expression of a Hox gene in R4, you're essentially changing the identity of that segment. It might try to produce the structures of R2 or R6.
That is exactly it. It's a system where positional information is encoded chemically. If you disrupt the Hox code, the migration paths of the NCCs are altered, and that leads to misplaced ganglia or improperly patterned cranial nerves.
Which would cause craniofacial defects.
Often, yes, because the rhombomeres also regulate the development of the pharyngeal arches.
So let's connect the specific rhombomeres to their cranial nerve motor nuclei.
The organization is highly predictable based on this segmentation. The motor neurons for the trigeminal nerve, CNV, are patterned in R2 and R3.
Right.
The motor neurons for the facial, CNSI, and abducens, CNSIV, are clustered in R4 and R5. And finally, the motor nuclei for the glossopharyngeal, CNAX, and vagus, CNX, nerves are found down in R6 and R7. The rhombomeres establish the functional segmentation that governs swallowing, chewing, and facial expression.
Okay, now let's move back caudally to the neural tube that forms the spinal cord. At stage 13, its wall is defined by 3 concentric layers or zones, which reflect the different stages of cell development.
These zones represent a progression of cellular life, from proliferation to differentiation. So innermost, lining the central canal, you have the ventricular zone, VZ. This is the master proliferative layer.
This is the factory.
This is the factory. All the non-neural cells, the glia, and all the future neurons are generated right here.
And then the cells that move out of the VZ, they settle in the next layer out.
That's the intermediate zone, or the mantle zone. As cells migrate out of that highly mitotic VZ, they enter the IZ, where they stop dividing and they start to differentiate into functional neurons. This zone is destined to become the gray matter of the spinal cord.
And the outermost layer.
That's the marginal zone, MZ. And that's fundamentally just composed of axons extending from the neurons that are located in the mantle zone. This axon layer will eventually become the white matter.
But the IZ, that future gray matter, it gets split into 2 functional halves. A dorsal half and a ventral half. What's the dividing line?
That internal division is marked by the sulcus limitans. It's this longitudinal groove that appears on the lateral wall of the neural tube, and it perfectly divides the gray matter into the dorsal portion, the alar plate, and the ventral portion, the basal plate.
And in the midline?
In the midline, you have 2 non-neural structures that cap the tube. The roof plate on the dorsal side and the floor plate on the ventral side.
And it's these non-neural midline structures that act as the master architects, controlling the fate of every neuron through signaling. This brings us to that crucial molecule, SHH.
The dorsal ventral axis patterning is just a beautiful example of gradient-based signaling. The notochord, that mesodermal rod lying just ventral to the neural tube, is the primary source of the signaling molecule SHH, sonic hedgehog. And the notochord induces the floor plate to also express SHH.
So SH originates ventrally at the bottom, and it diffuses upward, creating a gradient. What does that concentration gradient determine?
The concentration of SHH determines the cell's fate. High concentrations of SHH induce the cells nearest the floor plate to adopt a ventral fate, which leads to the development of motor neurons. That's the basal plate.
And the gradient itself is the key.
The gradient is the key. The sources detail that it dictates the specific motor neuron subtype. Extremely high SHH induces the floor plate itself. Slightly lower SHH induces the general somatic efferent, GSE neurons, the most ventral motor neurons. And a little higher up, a lower concentration induces the general visceral efferent, GVE neurons.
So the notochord isn't just saying be motor, it's giving precise instructions. Be this type of motor neuron at this specific location.
Precisely. And conversely, signals from the dorsal side, mainly bone morphogenetic proteins, BMPs coming from the surface ectoderm and the roof plate, they establish the dorsal fate, promoting the development of interneurons and sensory relay neurons in the LR plate.
So the whole spinal cord architecture is defined by these 2 opposing gradients.
The reciprocal gradients of SHH from the bottom and BMPs from the top.
Let's elaborate on the functional specialization of those 2 main gray columns, derived from the intermediate zone. Starting ventrally, the ventral gray column comes from the basal plate.
And this column is purely motor. It houses the cell bodies of motor neurons, which are organized based on the muscle type they're going to innervate.
So GSE and GVE.
Right. The general somatic efferent, GSE, neurons are the cell bodies that innervate skeletal muscles, the muscles from the somites, for voluntary movement. The general visceral efferent, GVD neurons, are the cell bodies of the presynaptic autonomic neurons, handling the involuntary control of smooth muscle and glands.
Then moving dorsally, the dorsal gray column comes from the alar plate.
This column is the sensory relay center. It contains interneurons and relay neurons that process all the incoming sensory information.
Yeah, right. And the sources categorize 4 main types of sensory input here.
GSA, SSA, GVA, and SVA. So that's general somatic afferent, GFA, for pain, touch, temperature from skin and muscle. Special somatic afferent, SSA, for specialized senses like vision and hearing, though that mostly terminates in the brainstem.
Okay.
Then general visceral afferent, GVA, for sensation from internal organs, and special visceral afferent, SVA, for taste and smell, which again is mostly rostral.
But it is vital to remember that the cell bodies for these sensory neurons are outside the spinal cord entirely.
Completely outside. They reside in the neural crest-derived dorsal root ganglia, DRG. The sensory fibers from the DRG send their central processes into the marginal zone and terminate in that dorsal gray column, forming the central relay stations for all sensory input.
We also have the lateral gray column, and its appearance is very restricted geographically.
It is. This column only forms in the thoracic and upper lumbar regions, generally from T1 through L2 or L3. Its sole function is to house the presynaptic neurons of the sympathetic nervous system, which is a GVE component.
So this is the physical location of the entire thoracolumbar outflow.
The defining characteristic of the sympathetic system. It's all right there.
Finally, let's talk about the maturation timeline, which really centers on that marginal zone becoming white matter.
As the neurons differentiate, their axons extend out into the marginal zone, and that forms the white matter tracts. But functional maturation depends entirely on myelination.
The process of wrapping these axons with myelin sheaths.
To dramatically increase the signal velocity, myelination is the cornerstone of neurological function, but it's a very long process.
When does this insulating process actually begin?
It starts surprisingly early during the second trimester in some specific evolutionarily critical tracks. The sources note that myelination proceeds in 3 distinct stages, starting at 3 different sites.
So spinal cord and brainstem first.
Yes, then the cerebellum, and much later in the cerebrum. The entire process continues through the neonatal period and well into adolescence. We monitor this closely with MRI postnatally because the degree of myelination directly correlates with a child's readiness to achieve certain neurological and motor milestones.
Okay, let's shift our focus back up to the rostral region, specifically the hindbrain derivatives, where that foundational spinal cord architecture gets dramatically modified by the pontine flexure. We'll start with the most caudal brain region, the myelencephalon.
The medulla oblongata.
Right.
The medulla is essentially the transition zone. It's positioned rostral to the cervical flexure, but caudal to that disruptive pontine flexure. The key derivatives here are the medulla oblongata itself, the caudal part of the fourth ventricle, and the choroid plexus of that ventricle.
How does the medulla maintain that motor sensory division of the spinal cord while also accommodating the physical spreading of the neural tube?
The pontine flexure is the key player here. In the spinal cord, remember, the motor or basal plate and sensory alar plate derivatives are stacked vertically.
Right, on top of each other.
But the flexure forces that alar plate to spread laterally, opening the tube up like a book. This physical change means the motor nuclei, which stay ventral, are now displaced laterally from the sensory nuclei.
And they organize into columns.
Distinct columns along the floor of the widened fourth ventricle.
So instead of that gray matter H shape we see in the cord, the nuclei are arranged in columns parallel to the midline, defined by their functional type.
Precisely. You find the motor nuclei, the GSE and GVE, clustered medially and ventrally closest to the midline. And the sensory nuclei GSA, GVA, SVA, SSA are progressively more lateral. That lateral displacement is the defining architectural difference between the brainstem and the spinal cord.
And it's a direct result of the pontine flexure opening the roof plate.
That's all it is.
Moving just rostral to the medulla, we find the metencephalon, which gives rise to the pons and the cerebellum.
The metencephalon derivatives are the pons, the rostral part of the fourth ventricle, and the entire cerebellum. The pons itself is derived from the basal plate and also elements of the alar plate. It's an essential bridge of fibers linking the cerebrum to the cerebellum and the spinal cord.
The pons houses the nuclei for a huge range of cranial nerves.
It does, yes. Motor and sensory nuclei for CNV, 6, 7, and some portions of ALV. The structural arrangement here is also defined by that pontine flexure, which stretches the roof plate to help form the superior medullary velum, and the telechoroidea, defining the roof structure over the fourth ventricle.
Right.
The pons is structurally complex. It contains all these ascending and descending fiber tracts alongside its unique cranial nerve nuclei.
While the pons is architecturally complex, the cerebellum is a complete biological masterpiece. It demands its own multi-step developmental sequence.
It is the most complex histogenesis outside of the cortex, for sure. The cerebellum, which is responsible for coordinated movement, balance, motor learning, it originates from the rhombic lip.
Which is the dorsalmost part of rhombomer 1.
Exactly.
Okay, let's dedicate some significant time to the cerebellum. In terms of just its external growth, how does it start to fold?
So the rhombic lip thickens, and it expands dramatically over the roof of the fourth ventricle. Initially, it forms the anterior and posterior lobes. But as growth continues, transverse grooves divide it into the hemispheres, the central vermis, that's the worm-like structure, and the flocculonodular lobe.
And the sources specified that the flocculonodular lobe is the oldest part, evolutionarily.
It is considered the most primitive part, crucial for basic balance.
The real insight, though, is in the internal layering, the histogenesis. It involves cells migrating in this bizarre inside-out, then back-inside pattern. Let's break down the 4 critical steps.
Step 1 is the initial cell production. Neurons are generated in the ventricular zone of the metencephalon, and these cells begin the process of migration.
And step 2 involves this massive outward migration to form a temporary surface layer.
This is the formation of the unique external granular layer, EGL. Cells migrate outward from the VZ and SVZ all the way over the pile surface of the developing cerebellum. And this EGL is intensely proliferative. It acts as the progenitor layer for the vast majority of the cerebellum's inhibitory neurons. We're talking the granule cells, which are the most numerous neurons in the entire brain.
The most numerous, period.
Period. And also the basket and stellate neurons.
That seems completely counterintuitive.
Yeah.
In the spinal cord, cells move out and they stay settled. Here, they move out, they proliferate on the surface, and then they move back in.
That brings us to steps 3 and 4. So step 3 involves the Purkinje cells. These are the main output neurons of the cerebellum, and they are generated very early by the VZ.
Okay.
They migrate outward, but they stop just beneath the EGL, forming this crucial single-cell thick layer, the Purkinje cell monolayer. They are the architectural scaffold for the entire structure.
So the core output cells settle first, and then this massive population of granule cells has to migrate past them.
Exactly. Step 4 is the granular migration. The granule cells, having been produced and matured in the EGL on the surface, must then migrate inward past the settled Purkinje cells to form the internal granular layer, IGL.
That is a staggering feat of cellular navigation.
It is. This massive inward migration is driven by molecular cues, including factors that are released by the Purkinje cells themselves.
So why does the cerebellum require this highly counterintuitive layering? Why migrate out, proliferate, and then migrate back in?
Well, the cerebellum is this massive computational machine that continues to grow and refine motor control pathways long after birth.
Right.
This architecture allows for a massive expansion of the granule cell population on the surface, in the EGL, to accommodate the immense functional needs of coordination, which continues to improve throughout infancy.
So the EGL sticks around for a while.
It does. The EGL persists, indicating active proliferation, until postnatal month 6.
That postnatal persistence has to be a key vulnerability marker.
It is. If the EGL persists beyond that 6-month window, or if the intricate migration of those early generated Purkinje cells is disrupted, it's strongly linked to neurological disorders.
Like what?
Abnormalities in Purkinje cell migration are associated with various forms of ataxia and neurological deficits, including an increased risk for cerebral palsy. It just highlights the non-negotiable precision of this developmental timeline. The functional circuitry of coordination is literally being built during that 6-month postnatal window.
We've established the hindbrain and the brainstem, the structures governing survival and coordination. Now we move to the prosencephalon derivatives, the telencephalon and diencephalon, the systems for higher cognition and regulation.
The diencephalon forms the deep central core of the forebrain. It's divided structurally by the hypothalamic sulcus, and its derivatives are essential relay and regulatory centers: the thalamus, the hypothalamus, the epithalamus.
Which contains the pineal gland.
Right, and the subthalamus.
Let's focus on the hypothalamus, that tiny but mighty regulatory center, and the formation of its associated master gland, the pituitary.
The development of the hypophysis, or pituitary gland, is a stunning example of two completely unrelated embryonic structures meeting and fusing to form a functional organ. It has a dual origin.
One neural, one oral. How do those two components interact?
The adenohypophysis, or anterior pituitary, develops from Rathke's pouch. And this is a highly significant upward invagination of the oral ectoderm, the roof of the embryonic mouth.
So it's growing up from the mouth.
Simultaneously, The neurohypophysis, or posterior pituitary, develops as a neural downgrowth, a stalk-like extension called the infundibulum, from the floor of the diencephalon.
So you have the oral ectoderm growing up and the neural tissue growing down, and they meet in the middle.
They meet, and the functional interaction begins.
Yeah.
Although the pituitary is physically formed and the pouch pinches off by stage 12, the critical endocrine function, the synthesis and regulation of hormones that's established later by stage 20, once the complex signaling between the neural and glandular components is stabilized. And that separation of Rathke's pouch from the pharyngeal roof, it leaves a small connection that usually disappears, but remnants can sometimes lead to tumors.
The diencephalon is also the origin point for the entire visual system.
The eye is truly an outgrowth of the brain. The optic vesicles appear as these lateral outgrowths from the walls of the prosencephalon.
Okay.
These vesicles then invaginate, they fold back on themselves, to form the optic cup, which differentiates into the retina. The optic cup then induces the surface ectoderm to form the lens placode, which also invaginates to form the lens.
And it stays connected.
The entire structure remains connected to the diencephalon via the optic stalk, which later becomes the optic nerve. This neural origin is why the retina is structurally considered part of the central nervous system.
And finally, the dorsal structures, the thalamus and epithalamus.
The thalamus swells massively from the lateral walls of the diencephalon, becoming the major relay center for nearly all sensory and motor information heading to the cortex. Meanwhile, the epithalamus forms from the roof plate. It gives rise to the pineal gland, or epiphysis, which is a diverticulum of that roof plate, and plays a key role in regulating circadian rhythms through melatonin production.
We now turn to the telencephalon, the largest and most complex derivative, destined to become the cerebral hemispheres, the seat of complex human consciousness. And it starts simply as just two lateral buds.
Initially, that's all it is. The telencephalon is just two lateral swellings, the cerebral hemispheres, connected across the midline only by the thin lamina terminalis.
And their growth is explosive.
Explosive. They expand forward, creating the frontal lobe, upward, backward for the parietal and occipital, and downward for the temporal lobe, ultimately enveloping the diencephalon and mesencephalon entirely.
Functionally, the telencephalon is quickly divided into the dorsal pallium and the ventral subpallium.
The pallium is the dorsal region, which will differentiate into the six-layered structure of the cerebral cortex, the neocortex. The subpallium is the ventral region, and it's destined to form the crucial basal nuclei, the caudate nucleus, the putamen, and the globus pallidus.
Let's dedicate some time to cortical histogenesis. The formation of the cortex is a defining characteristic of advanced brains, and it requires incredible organizational precision, proliferation followed by radial migration.
The entire cortex is built from the inside out. It's based on a massive output of cells from two deep proliferative zones. The ventricular zone, VZ, lines the lateral ventricles, and just outside it is the subventricular zone, SVZ. These two zones are the proliferative engine room, just churning out billions of neurons and glial precursors.
And these cells need guidance to travel from the ventricle surface all the way to the outer pile surface. They need a path.
They rely on specialized cells called radial glia, and these cells serve a dual function. They act as progenitors themselves, but crucially, they also serve as fixed scaffolding.
Like train tracks.
Exactly like train tracks, extending from the VZ all the way to the pile surface. The migrating neurons essentially grab onto these glia and climb toward their final destination.
And this movement follows the strict inside-out migration rule.
This rule is paramount for establishing the 6 distinct functional layers of the cortex, layers I through 6. The first neurons generated in the VZ migrate outward, and they settle the deepest, forming the future layer 6.
Okay.
The second wave migrates past the first wave to form layer 5, and so on. The latest generated neurons, those destined for the most superficial layers like layers 2 and 3, the associational layers, they have to migrate the furthest, passing through all the previously settled layers.
And this precise layering ensures that the deep output layers are built before the superficial integration layers.
It does.
Within this complex layering process, the sources highlight a critical but transient structure. Subplate zone. Why is this temporary zone so important?
The subplate zone is located just beneath the rapidly forming cortical plate. It's a temporary holding zone. The very first neurons to migrate, even before layer 6 is fully established, settle here.
And it peaks around 26 weeks.
Yes, its density peaks around 26 weeks gestation. Its function is to serve as a transient synaptic way station. Thalamic axons, which are projecting sensory and motor information to the cortex, they pause and they synapse in the subplate before they can reach their final target layers in the fully formed cortex.
So it's essentially a temporary connection center that allows the brain to start wiring and testing circuits before the final cortical structure is even complete.
Exactly. It guides the establishment of intracortical and thalamic connections. And once the 6 layers are mature, the subplate cells largely disappear through programmed cell death.
And if it's disrupted?
Disruption of the subplate is thought to be involved in major neurodevelopmental issues, as it compromises the brain's ability to establish those core sensory relay pathways.
The excitatory projection neurons are formed in the pallium and migrate radially. But we also have this massive population of inhibitory neurons that are formed elsewhere and migrate tangentially. Where do the basal nuclei and these inhibitory interneurons come from?
They both originate from the subpallium, specifically from the medial and lateral ganglionic eminences, MGE and LGE. The LGE is the primary source for the differentiation of the deep basal nuclei, the caudate nucleus, the putamen, and part of the globus pallidus.
And the MGE handles that tangential migration.
Correct. Cells generated in the MGE migrate tangentially sideways across vast distances, traversing the intermediate zone and spreading throughout the developing pallium.
And these become the inhibitory neurons.
These cells differentiate into the deep cortical interneurons, which are primarily GABAergic or inhibitory. This tangential migration is essential for providing the cortex with the necessary inhibitory circuitry to balance the massive excitation, ensuring normal function and preventing seizures.
The massive telencephalon needs its 2 halves to communicate. This requires the commissures, these huge fiber bundles crossing the midline. And the location where they all converge is the lamina terminalis.
Yes, the thin rostral end of the neural tube, the lamina terminalis, acts as the physical gateway through which all the major commissures have to cross the midline. This is essential for the coordinated function of the 2 hemispheres.
So which is the first major commissure to form?
The anterior commissure forms first. It connects the olfactory bulbs and portions of the temporal cortices, mediating some non-olfactory communication between the temporal lobes. After that, the hippocampal commissure develops, connecting the hippocampal formations via the fornix system, which is crucial for memory pathways.
And then the massive highway that defines the human brain structure, the corpus callosum.
The corpus callosum, CC, is the largest commissure by far. It connects the neocortices of the 2 hemispheres. It arises as this thick, continuous fiber mass from the area of the lamina terminalis.
And it grows rapidly.
It starts forming around 10 weeks gestation and then rapidly expands backward, forming its distinct parts. The rostrum, the genu, the trunk, and the splenium. Its rapid, enormous growth just reflects the exponential expansion of the cerebral cortex itself.
And if that complex crossing mechanism fails, the results are profound. It leads to a genesis of the corpus callosum.
If the CC fails midline, they can't find their path.
So where do they go?
Instead, they form these aberrant, misrouted bundles that run parallel to the midline within the medial walls of the lateral ventricles. These are known as Probst bundles.
And a genesis is associated with cognitive deficits.
Varying degrees of cognitive and functional deficits, because the left and right hemispheres just cannot share complex information effectively. It's a clear demonstration of how a failure in architectural pathfinding leads directly to functional separation.
We've built the CNS, the central command. Now we return to the periphery to see how all the connections are established, starting with the simplest structure, the spinal nerves.
Spinal nerves are the most fundamental composite structure of the PNS. They're formed by the union of 2 roots. You have the motor roots, which carry efferent, or output, axons originating from the motor neurons in the ventral spinal cord.
From the basal plate.
From the basal plate. And then the sensory roots, which carry afferent, or input, fibers originating from the neural crest-derived dorsal root ganglia.
So the architecture demands that the motor output grows out to its target and the sensory input grows in to connect to the CNS relay centers.
That's it exactly. The motor fibers exit the basal plate and pierce the meningeal layers to innervate their target muscles. Simultaneously, the sensor fibers from the DRG penetrate the marginal zone of the neural tube, forming the dorsal columns that convey sensory information up to the brain. The whole system is elegantly segmented, reflecting the sommite structure.
The cranial nerves, I through 12, are the functional extension of the CNS into the head and neck, but they are dramatically more complex. They often contain multiple fiber types and have dual origins.
The fiber classification is extensive. You have motor types like GSE and GVE, specialized motor or SVE, and then all the various sensory types, GSA, GVA, SVA, SSA.
And their nuclei in the brainstem, they follow that spinal cord rule of ventral motor, dorsal sensory.
They do, but as we noted, it's all spread laterally by that pontine flexure.
Let's nail down the composition of the ganglia, because that's the most confusing element. Which nerves have a purely placode origin?
The sensory components of the special sense nerves are primarily placode derived. So that's olfactory, CNI, optic, CN2, which is really a CNS tract, and vestibulocochlear, CN8.
Right.
The sensory neurons for hearing and balance in CN8, they come from the otic placode.
And the composite nerves, where the neural crest and placodes share the duties.
This includes the sensory ganglia of CNV, 7, 9X, and X. For example, the trigeminal ganglion for CNV gets its GSA component from the neural crest, but it has contributions from placodes as well.
And the vagus nerve?
Similarly, the sensory ganglia for the vagus, CNX, they get their GVA and SVA components from the epibranchial placodes, and that ensures specialized visceral sensation.
And finally, the specialized motor nerves, the branchiomeric nerves. What makes their motor component unique?
The branchiomeric nerves, which are V, 7E, X, and X, innervate the muscles that are derived from the pharyngeal arches. So think muscles of mastication, facial expression, the pharynx.
Okay.
They contain special visceral efferent, SVE, fibers. Now, although they're named visceral, they are functionally somatic. They control striated muscle, but their unique embryonic origin from the arches gives them this distinct classification and placement within the brainstem nuclei.
The vast involuntary control system, the ANS, is a massive testament to the capability of neural crest cells.
The ANS relies almost entirely on the neural crest for its peripheral components. NCCs form all the postganglionic neurons for both the sympathetic and parasympathetic divisions, and all those specialized neuroendocrine chromaffin cells of the adrenal medulla.
Let's contrast the two divisions, starting with the sympathetic nervous system, the thoracolumbar outflow.
The presynaptic sympathetic neurons, the GVEs, originate exclusively from the lateral gray column of the spinal cord, from T1 to L2, L3. The NCCs migrate ventrally and differentiate into the ganglia.
Okay.
Crucially, the paravertebral ganglia form the sympathetic trunk, which aligns segmentally alongside the vertebral column, maintaining that strict segmentation we talked about. The prevertebral ganglia form further out, like the celiac plexus.
And the architecture is short preganglionic, long postganglionic fibers.
Exactly. And the parasympathetic nervous system, the craniosacral outflow, operates completely differently.
How so?
Its presynaptic GVE neurons originate either from specific brainstem nuclei, so cranial nerves 3, 7X, and X, or from the sacral spinal cord S2 to S4.
And the NCCs have a longer journey.
A much longer journey. The NCCs destined for this system undertake long-distance migration, forming ganglia near or even within the effector organs. We call these terminal or intramural ganglia. And this architecture results in long preganglionic and very short postganglionic fibers.
This long migration leads directly to the formation of the enteric nervous system, ENS, the so-called second brain.
The development of the ENS is one of the most remarkable examples of long-range NCC migration. The vast majority of the ENS, which controls gut motility and secretion, is derived from two populations of NCCs.
Which are they, and how do they colonize the gut?
The primary source is the vagal neural crest, from cranial nerve X. These cells migrate extensively from the level of the hindbrain, travel down into the foregut, and then they colonize the entire gut tube in a strict rostral to caudal wave.
A wave of cells moving down the gut.
A wave. Our sources highlight their incredible motility, measured at roughly 40 micrometers per hour in development models, as they navigate through the gut mesenchyme.
And the second population helps finish the job at the distal end.
Yes, the sacral neural crest cells colonize the distal colon. The two streams meet and merge to form the entire intrinsic nervous system of the gut.
So if the vagal NCCs fail to complete their long migration, what is the clinical result?
The result is Hirschsprung's disease. This is defined by the failure of the NCCs to successfully colonize the distal segment of the gut. This results in a ganglionic segment, a portion of the intestine that lacks the necessary nerve cells, the ganglia, for paracelsus.
Which causes a functional obstruction.
A functional obstruction and severe chronic constipation. It directly links a failure of embryonic migration to a severe postnatal gastrointestinal disorder.
We've established the neural architecture. But to maintain and feed this rapidly growing organ, We need a robust, resilient blood supply.
The arterial supply begins very early.
Yeah.
It's sourced from branches of the dorsal aortae and the third aortic arches. And we distinguish the two critical circulations that feed the entire system.
The anterior circulation is based on the internal carotids.
Correct. The anterior circulation derives from the internal carotid arteries, and it supplies the cerebral hemispheres via the middle and anterior cerebral arteries. This handles the large, rapidly expanding forebrain.
And the posterior circulation handles the brainstem and hindbrain.
The posterior circulation is derived from the vertebral arteries, which merge to form the basilar artery, supplying the hindbrain and cerebellum via the cerebellar arteries. And these two systems have to be linked for redundancy.
That link is the famous Circle of Willis.
The Circle of Willis. It's a crucial anastomosis of leptomeningeal arteries at the base of the brain, linking the anterior and posterior circulation systems. The sources specify that this circle is typically completed and functional by 20 postmenstrual weeks.
And that establishes a vital safety mechanism.
It provides collateral blood flow in case of a proximal vessel obstruction.
How does the vasculature grow into the solid tissue of the brain itself?
Vascularization happens primarily via angiogenesis, that's the sprouting of new vessels from existing ones, rather than vasculogenesis, which is forming vessels de novo. Deep vessels, what we call ventriculopetal arteries, supply the ventricular zone, while superficial vessels, the leptomeningeal arteries, supply the cortex surface.
Yes. And this vascularization leads us to the critical, highly vulnerable structure of the premature infant, the germinal matrix.
The germinal matrix is a transient, highly proliferative, and highly vascularized layer located in the VZSVZ of the telencephalon. Its vasculature is unique.
How so?
The vessels are immature. They lack sufficient structural support parasites and astrocytes around their very thin walls, and this fragility persists until about 34 weeks gestation.
This inherent fragility makes premature infants highly susceptible to what condition?
To intraventricular hemorrhage, or IVH, any fluctuation in blood pressure, which is common in premature labor, can cause these fragile vessels in the germinal matrix to rupture, leading to bleeding into the ventricles.
Which is devastating.
It's a major, devastating cause of neurological deficit in preterm infants because the GM is both a cell factory and a massive vascular liability.
To protect the delicate neural environment, the brain has to tightly regulate what crosses the blood vessels. That's the function of the blood-brain barrier.
The BBB is far more than just a wall. It's a dynamic, functional barrier that controls the passage of substances from the blood into the CNS. Its formation requires the precise assembly of what we call the neurovascular unit.
Which involves endothelial cells, parasites, astrocytes,
and neurons, all working in concert.
What gives the endothelial cells their special impermeability?
They develop incredibly tight junctions, far tighter than capillaries anywhere else in the body, and they express specific transport proteins that actively control what is allowed to pass. This highly regulated exchange is essential for maintaining the chemical homeostasis required for neuronal function.
And we have to distinguish this from the CSF barrier.
Yes. The blood cerebrospinal fluid barrier, BCSFB, is anatomically separate. It's formed by the specialized epithelium of the choroid plexus, which actively secretes CSF into the ventricles. Both barriers serve protective functions, but the BBB protects the brain parenchyma itself, while the BCSFB controls the fluid environment of the ventricles.
And how does the maturity of the BBB relate to neonatal vulnerability?
The barrier becomes progressively less permeable as gestation proceeds. In premature infants, the BBB is often reduced and less effective.
So it's leakier.
It's leakier, and this compromised protection makes them disproportionately vulnerable to systemic infections, to toxins, to large molecules that would be blocked in a term infant. It allows systemic insults to directly affect the developing neural tissue.
Looking at the brain of a term neonate, structurally and functionally, where is it in terms of completion?
At term, the brain weighs about a quarter of the adult weight. Structurally, the cerebrum is disproportionately large compared to the cerebellum, which still has a lot of growth to do postnatally.
The major architectural folding, the sulci and gyri, is well underway, but it's not finished.
Folding begins around 24 weeks gestation, and the major sulci continues postnatally, especially the smaller tertiary sulci, as the cortex continues its massive surface area expansion and maturation.
We discussed the vulnerability of the germinal matrix before 34 weeks. By term, that vulnerability is largely gone, correct?
Correct. The highly proliferative, highly vascular germinal matrix disappears almost completely by 40 weeks gestation. And this is the physiological reason why term infants are at a vastly lower risk for IVH compared to preterm infants.
But the white matter itself has another major vulnerability in prematurity: periventricular leukomalacia, or PVL.
PVL is specific damage to the white matter, the leukomalacia, in the area immediately surrounding the ventricle, so periventricular. This region, which contains the developing axons of the corticospinal tracts and other major projection fibers, lies in a watershed area of the developing blood supply.
A zone where blood flow is marginal.
Very marginal. And premature infants who lack robust autoregulation are extremely sensitive to poor oxygen or blood supply to this area. PVL leads to necrosis and cystic changes in the white matter, and that results in permanent neurological deficits, often resulting in cerebral palsy or cognitive impairment.
Shifting to function, what reflexes that are present at birth confirm the functionality of the lower CNS centers?
The presence of these neonatal reflexes confirms that the spinal cord and brainstem are fully organized and operational. Key examples include the rooting reflex, the Moro reflex, or startle reflex, and the incredibly complex suck-swallow reflex,
Which requires a dozen different nerve components.
a coordinated action of 12 different cranial nerve components from V, 7, 9, X, and 12.
And the Babinski reflex is the classic marker of the CNS level of organization.
The Babinski reflex, the extensor plantar response, where the toes fan up, is normal in newborns. Its presence indicates that the corticospinal tracts, the higher inhibitory pathways, have not yet fully myelinated and established inhibitory control over the lower spinal cord reflexes.
So its disappearance is a key milestone.
Its disappearance within the first year is a critical developmental marker, indicating the maturation and functional control of the higher brain centers.
Finally, the long, slow process of maturation that defines the postnatal development of the brain: myelination.
Myelination, the final step in optimization, continues throughout adolescence. It is the process of wrapping axons to increase the velocity and the efficiency of signal transmission, and it proceeds chronologically based on functional importance.
What's the timeline and the functional progression?
Myelination starts early in the brainstem, the cerebellum, and the sensory and motor spinal tracts, because those are essential for immediate survival and movement.
Right.
In the cerebrum, the process moves from the primary sensory and motor areas and gradually progresses to the higher order association areas like the frontal and temporal lobes. The sources provide a detailed timeline showing that the myelination of tracts supporting complex cognition and planning is among the very last to be completed.
Confirming that the brain is not finished wiring until the late teens or early 20s.
That's right.
Which means the brain isn't just growing. It's functionally wiring itself for speed and efficiency for decades after we leave the womb. What an incredible journey we've taken today, tracing the development of the nervous system. We started with a simple ectodermal sheet at 18 days. We mapped its transformation into a tube governed by these complex signaling gradients like SHH.
Right.
We tracked the massive migrations of the neural crest building the entire PNS, and we detailed the sophisticated inside-out layering of the cerebral cortex. It's an architectural marvel built on immense precision and non-negotiable timing.
The detail we explored today really highlights that development involves a sequence of fragile, transient structures: the EGL in the cerebellum, the highly vascularized terminal matrix, the synaptic subplate zone. These structures are essential, but their temporary nature makes them specific targets for developmental failure or injury.
Leading directly to conditions like Hirschsprung's, IVH, or neurological delays.
Exactly. Understanding these transient phases is key to grasping the full scope of congenital and premature vulnerabilities.
We saw that the core anatomical framework, the 6 cortical layers, the basal ganglia, the brainstem organization, is largely established within very specific windows of time in utero. However, the final piece of functional architecture, the myelination that optimizes these connections, continues well into late adolescence.
And that raises an important question for you, the learner, to consider as you reflect on this journey. If the process of myelination, the optimization of neural pathways for peak performance and speed, continues through the high plasticity period of adolescence, and it governs the final efficiency of cognitive and association areas, what subtle cognitive functions might still be structurally and functionally vulnerable to environment, to stress, to learning or experience during those crucial later stages of growth?
And how does that extended window of refinement fundamentally influence our understanding of personality development, learning retention, and behavioral plasticity in young adulthood?